Hello and welcome to today's webinar organised by Thermo Fisher Scientific. Good afternoon, good morning, good evening, wherever you are joining us live or on demand. Thank you for joining us in celebrating 50 years of ion chromatography innovation. And my name is Wai-Chi Man and I am an IC Product Marketing Manager. Our presentation today is what can ion exchange chromatography with mass spectrometry bring you to metabolomics, which will cover how ICMS plays a role in the world of omics science. Our speaker today is Professor James McCullagh from University of Oxford in the UK. Before our presentation begins, we would like to mention a few tips that will enhance your webinar experience. So please submit your questions at any time during the presentation in the Ask a Question widget pinned in your console. We value your time and want to ensure that we deliver high quality and relevant content. Please use the survey to share your comments and suggestions. Explore the menu dot along the bottom of your screen. Those buttons open and close all the widgets and windows. Lastly, we are recording today's webinar. The on-demand link will be sent to all the attendees via email approximately 24 hours after the broadcast. Thank you again for joining us today. Our presentation will now begin. So take it away, Professor McCullagh.
Thank you very much, Wai-Chi, for that introduction. And also thank you to Thermo for inviting me to talk today at this webinar. So what I wanted to do was to really talk a little bit about how we've got involved in ion chromatography coupled to mass spectrometry and utilize that for metabolomics in particular, and how that's kind of fed into our multi-omics and our omics science platforms. And I just want to take the opportunity really to sort of talk a little bit about the method development that we've done in that context, but also some of the applications that we've been working on over the last sort of 10 to 12 years, actually. So just quickly before we get started, my disclosures that I do work with in collaboration with Thermo Fisher Scientific. We have a research collaboration around ICMS, and I also have a DPhil student who is supported by Thermo Fisher Scientific as well. So just a little bit of background about our research group and what we do. So we really revolve around using mass spectrometry and separation science for a range of different applications, particularly omics science applications in systems, and to understand systems biology, to understand the small molecules in particular, but the interaction with large molecules too, the chemical systems that take place in biological context. We're very interested in cellular function and using omics science to do that. So we use a range of techniques, but ion exchange chromatography has become one of the preeminent tools in our lab, as you'll see. So I guess there's sort of three pillars of what goes on in our lab. One is technology development around what we'll talk about today and in some other areas as well. And then applying that in an omics science kind of context to understand biological systems. And then use that information, and what we try to do is use that information to be to look at mechanisms and biomarkers, and to use a sort of systems approach to understanding and developing new therapeutic targets, and indeed the impact of therapeutics on those biological systems. We believe that there's a lot of information to be gathered from understanding the impact of disease states, for example, on cellular metabolism, and indeed the impact of drugs to treat those diseases on that system. And that's really where we focus our time. So just to sort of encapsulate that, we really go from features, things that we can measure in a mass spectrometer that are signals, and try to move that through a process or a pipeline to understanding function and within biology. So that's what we do. A lot of what we do, as I say, has been kind of directed by the last 10 years' work on ion exchange chromatography. And this is just a brief timeline from 2013, when we received our first instrument, through to today. And I want to sort of take you on a journey in this webinar, I guess, through that process, and sort of highlight two areas, one being how we've developed the methods and validated those methods, and then secondly, where we've used those and what has driven that method development in a way, what's inspired that method development in terms of how we've applied that to biological questions that we've been asking. So we started 2013 and been working ever since then, actually, with ICMS. So just to go back to 2013 and just introduce this work through some of the research, well, the research question that really started things off. And it was through a collaboration that sort of grew, and these people here are those that were there at the beginning. And it was really around trying to understand cancer metabolism in a particular context associated with isocitrate dehydrogenase mutations that were found in glioblastoma cells. And this was a discovery. I'll give you a brief background, because this drove a lot of our initial ICMS development work. So initial discoveries by others in 2008 showed that mutations in isocitrate dehydrogenase were prevalent in glioblastoma. And then in 2009, and this was key and where we became interested, it was shown that these mutations lead to a change of function and the production of a metabolite called 2-hydroxyglutarate. And this was an unusual mutation because it was a change of function, not a deletion of function. And the accumulation of 2-hydroxyglutarate was clearly occurring in glioblastomas and gliomas at this point. But it was unclear as to what that was doing, why that was there. Subsequently, it was found that these mutations are actually found in quite a few different types of cancers, gliomas, but also acute myeloid leukemias, chondrosarcomas, bone cancer, cholangiocarcinoma, and others. And in fact, there is a list where of cancers that's nearer 20 now that have found these mutations in these cancers. Now, the interesting thing about them is that they are early mutations. They happen very early in the tumor development process, and therefore are driver mutations. They are clearly helping that tumor develop. So our questions were really at this point, well, what is that process? Why are these mutations helping tumors develop early on? And what's the impact on metabolism, which is where we were interested. So yeah, just sorry, forgive me, sort of zooming in on biochemistry here, but just to see where that IDH, those IDH enzymes are prevalent. They're found both in mitochondria and the cytoplasm of cells, and the isocitrate dehydrogenase wild type is a natural enzyme that converts isocitrate to 2-oxyglutarate. Sorry, I don't have a pointer, but you can see here in the TCA cycle, isocitrate on the right-hand side, being converted to 2-oxyglutarate. But the mutant and the mutant enzyme, the change in function for the mutant enzyme is that it converts 2-oxyglutarate to 2-hydroxyglutarate, just below that. So 2-hydroxyglutarate is a natural metabolite, but it accumulates to unusually high levels in these tumors. There's also a cytoplasmic version of IDH as well, which is also mutated or can be mutated in these tumors. But interestingly, they're mutually exclusive. You don't see both mutations. You only see one or the other in tumors. Okay, so that was the sort of background. We wanted to find out what's the impact of these mutations in tumors or in tumor cells. So and the relevance of the metabolism aspect of this, of course, this is a mutation that is directly at the heart of metabolism, clearly from, as you can see here, from the TCA cycle. So it must be affected. Well, the hypothesis was it would be affecting metabolism, which would be supporting tumor genesis. Okay, so how do we do that? So we immediately started to think about methods for analysis of metabolism, and this is where things became more tricky. Probably the most common method for analysis of central metabolism would be HILIC MS. It's been around for a long time. But we really had trouble with this when we were trying to look at central metabolism. And indeed, if you look at the literature, it is not a one method fits all central metabolism approach. In fact, and Centropar, this picture here shows it's particularly difficult for highly ionic metabolites, such as the phosphorylated, as well as nucleic acids, which is highlighted here as being particularly difficult to characterize. We were just finding it was very difficult to get reliable, reproducible, comprehensive coverage of primary metabolism, which is what we were trying to do. So going from that difficult methodology to understanding the biological system was a problem. We went through various approaches. We looked at iron pair as well. Forgive picture here. This is just the earliest picture I could find of our iron chromatography system, because at the time we bought a Q Exactive, and Thermo provided us with a seed IC system, having had discussions with them about our challenges with HILIC. And so we had that installed alongside our Q Exactive. And just in the background behind there is our system that we did. It's a U3000 system that we were doing our HILIC on as well as reverse phase. So we were trying to take a multi LCMS approach here. First ICMS 2008. And the approach we took was to try to benchmark or compare HILIC MS with AECMS. This is ion exchange chromatography coupled to mass spectrometry, but in most of what I'll talk about today, we were in anion exchange, looking at negatively charged ions, negatively charged polarities. So we call it an exchange to distinguish from cation exchange. We plugged both of them into our Q Exactive and then started to do some comparisons. So there are sort of four parts to my talk. I probably should have said at the beginning. And so we're on to the second part, which was really developing the methodology for ICMS. There's some people here who have been instrumental along the way. There are others, but these were those early on. So just to give you a bit of background to ICMS and what it does. So ion exchange, we're all familiar with ion exchange chromatography, and this is ionic interactions that take place on a charged resin column, and then elution for depending on the polarity, on whether it's anion or cation. For anion exchange, elution is often with hydroxide ions that then replace your analytes that have been interacting with the opposite charge on the column. Or with cation exchange, protons are often used to do that exchange as well. And so the system that we had, ion exchange has been around since 1975. We're celebrating 50 years. Modern ion exchange with ion suppression technology has. And the system that enables us to couple to a mass spectrometer has two really important aspects I wanted to highlight. It's an ion exchange chromatography system with a column, etc. But the first thing that's important is this eluent generator. And this is where the hydroxide ions for anion exchange are generated. And the key thing here is that this is a gradient, in our case, a hydroxide ion gradient to resolve as many of the metabolites as possible. This is generated online from a concentrated solution that you add to the system maybe once a year. And it's a cartridge that you replace. And all you therefore add is purified water, millicube water to the system. So there's no making up each time you want to do analysis, mobile phases. And this turns out to be really important for metabolomics, where you want reproducibility is a key aspect, as we'll see. So that's one part. And the other one is the electrolytic ion suppressor, which really is the key to be able to couple a chromatography system that essentially has high hydroxide or proton content in the mobile phase, which is not something that is compatible with electrospray ionization, which would in the mass spectrometer. And so what we need to do is remove those eluent the hydroxide or the proton. So hydroxide for anion exchange is removed by electrolytic production of protons and hydroxide ions in this suppressor. And the protons are then used to neutralize the hydroxide in the mobile phase to produce water. And then semipermeable membranes remove the counterion, the potassium in this case usually, from the system. And so what you end up with at that point is having performed ion exchange is resolved analytes in aqueous solution, which is very suitable for then introducing to electrospiralization mass spectrometry. So that's how that works and how the system works. And it works very well. And key, it's robust and reproducible. And this is something that we found very quickly to be particularly helpful in an omics science context. So what we did was to then take that platform and to start having a look at metabolites. In the end, we screened over 400 metabolites, a few of them listed, just to show these are metabolites you'd find in mammalian cells. We also looked at some other biological contexts as well. And this is a picture. This is an early chromatogram from early days where we were running these standards as different mixtures. I think there's a couple of hundred metabolites in this mixture, and just shows over a long run, this is 60 minutes, that we get we get elution based on ionic strength, essentially. So high ionic strength, diphosphates, triphosphates will elute late, less ionic strength will elute early on. And actually, this is really helpful because you can really predict quite straightforwardly where you think a structure will elute, and it often, not always, does. And so therefore, that predictability is also a help when it comes to identifications. 60 minutes is a long run. We actually run a 37-minute run now, but just to look at everything, we had a long run going. So 400 metabolites, actually it's a bit more than that now, probably nearer 500, but that's what we published. And this is published work, so we've shown this in our communications biology paper back in 2020. So we did a head-to-head, as I say, against Hillick. And I mean, one of the things that came across quite straightforwardly, quite quickly when we were doing validation of that benchmarking, was the slope of the curve for the linear response to metabolites that we find in central metabolism. So I've got 2-hydroxyglutarate up there. There's a few examples here, and they're just highlighting different structures, really. But the slope of the curve, steeper for ICMS for all of these. Not always. There are a few metabolites that don't, particularly the less ionic strength metabolite, the lower ionic strength metabolites will not necessarily have such a difference. But this essentially shows us there's more sensitivity to the analysis for ICMS than Hillick for these metabolites. And so linearity and sensitivity were really good. The other thing that we were really struck by was the retention time reproducibility comparison for the same metabolite or some selection of similar metabolites for Hillick versus ICMS. And those who do Hillick would probably recognize that it's not always so reproducible. And that is one of the problems when you're applying it to for discovery science, where you need to really be sure of identifications, be sure that you're comparing like for like between different sample matrices or types or time. And so you can see the step change in reproducibility for ICMS there. And that really translates through in omics science context to greater confidence in our discoveries. And then, finally, just a few. These are just spot checks that we looked at many metabolites, but just comparing the low limits of detection in the low nanomolar range for most of the metabolites through including these tricarboxylic, dicarboxylic acids found in the TCA cycle, for example, and a range of the retention time range in seconds actually that's represented on that bar graph as well. So we're talking less than 10 seconds over 18 days, 18 injections over 18 days for the ICMS. So yeah, we were really happy with our validation and the robustness and the benefits that over Hillick that we were getting from this. Just a quick look here, in particular focus on nucleotides that were highlighted earlier on by Centropoir as being difficult by Hillock. We can resolve those very nicely. In fact, we can move those around on our chromatogram based on the gradient quite well. For ICMS, Hillock struggles with the retention of these triphosphorylated monodi and triphosphorylated nucleic acids as they are. Okay, and then one other example, one key example really, when you're looking at central metabolism, particularly energy transduction, which is implied through that pathway of TCA cycle through and glycolysis beforehand. A lot of phosphorylated sugar intermediates involved in those processes, and to understand, and branching points too. So the pentose phosphate pathway branching from glycolysis, for example, and also branching points around the TCA cycle. There are 22 sugar monophosphates that all share the same chemical formula, right? Isobaric in the mass spectrometer. So the chromatography to separate those is really important to get that sort of fidelity around central metabolism. This actually is probably not the best example, probably got fancier chromatograms, but it does illustrate that with the ICMS system, we can really separate out these monophosphates that all have the same chemical formula nicely across time with the ICMS system. And it turns out that this is really important when it comes to energy transduction and picking up differences in that flux through glycolysis and the pentose phosphate pathway. And one last example, just looking more at a sort of whole cell example of differences between some of these methods. And this is these are cell lysates being analyzed by ICMS, Hillock, and CAT in reverse phase. And this is has more of a biological focus to it. These are 4 different sample types, 2 of which are the mutant and the wild type IDH that you find in these in the cancers I've introduced. But we've also grown those cells at 2 different glucose concentrations as well. And you look on the left-hand side, you can see how ICMS has resolved those genotypes and the treatment concentration groups very nicely, and the QC samples that are pooled from those 4 are nicely centered between them. So we can resolve those well. These are PCA plots, principal component plots. Whereas Hillock, on the other hand, has got nice QC clustering and the ability to separate the 2 different glucose concentrations quite well on the left and the right. But the overlapping genotypes suggest that its fidelity to resolve the impact of IDH mutations is less good. And that's got clearly implied by the overlap of those circles. So interestingly, CAT in reverse phase was fairly good at being able to do this too. But for reasons, metabolite reasons other than those picked up by Hillock and ICMS as it happens, because we don't resolve or retain 2-hydroxyglutarate, for example, or many of those primary metabolic processes. Metabolites found in primary metabolic processes such as the TCA cycle. So this would imply that there are more hydrophobic metabolites that are also driving, can show difference between both the mutant wild type, but also on different glucose concentrations as well. So a lot of information inherent here in these sorts of PCA plots. Oh, I should have put that up. That would have helped you distinguish the groupings. But as I said, mutant and wild type cells grown on 20 millimolar glucose and 5 millimolar glucose separately. We did publish this work too, so this is, you can go and see this in the 2020 Communications Biology paper. More recently, we have published our anion exchange chromatography method for the analysis of cells, tissues and biofluids in Nature Protocols. Came out this year, and that is really a refined version of the method that was in the 2020 paper, with some more workflow for omics analysis, for metabolomics in particular, and taking the analysis all the way through from sample preparation through to data processing and analysis as well. Okay, so part three, here I wanted to move towards, so having validated the ICMS methodology, we started to look at the biology of these IDH mutations found in tumors. And so just wanted to take through some of our findings, so the application of ICMS in metabolomics itself. And several people have been involved, others ongoing. This is a long-term project we have. We want to understand IDH mutations and ideally find therapeutic, effective therapeutic interventions. And so we have ongoing work as well. Yeah, a few students who are currently in the lab here, as well as past students who've contributed. So just to set the scene for some of these applications, we're doing cellular investigations, looking at cells in tissue culture. And these are glioblastoma cells that we're going to look at here, but there are other cell lines that we use as well. The glioblastoma cell lines that have got the mutation inherent, and also those that are the wild type as well. So we're really focusing on what's the impact of the IDH mutation itself on cellular metabolism. And we take cells, in this case, we'll be looking at 9 replicates of tissue culture grown cells, harvested. We essentially make it using methanol as an extraction solvent, removing protein and large molecular weight metabolite compounds, non-metabolites, and retaining metabolites for analysis. And so that's our workflow. A little bit more detail here. This can be found in our publications as well. But it's essentially a conventional metabolomics workflow from sample through to data analysis. The only difference being that we're utilizing ICMS instead of other LCMS approaches. But it works in very much the same way. So when we do that comparison between the mutant and the wild type cells, one of the things that's clearly apparent when we look here, this is a volcano plot of the analysis, the experimental conditions here, is that we're seeing lots of changes in compounds beyond 2-hydroxyglutarate. I have purposely shown here the untargeted analysis. So I've named a few metabolites on here, but many of these have not been identified yet. Or we have identified them, but I'm just putting up the untargeted data here. You can see some of the data analysis in general. So this is analysis was from early on. We had 3,500 compound features measured by ICMS. About 2,000 of those had a CV percentage CV value less than 30, so reproducible for untargeted analysis. And we identified about 164 of those metabolites using our database of authentic standards that I mentioned earlier on. And then started to look at some of the significantly altered compounds. And so there were quite a few. Here's just a small selection of those where we saw significant events, so changes in the mutant cells. 2-hydroxyglutarates on the top left, and then several other metabolites that have gone down in abundance, or indeed there are a couple that haven't changed that are notable. For example, citrate hasn't changed in abundance, although its proximity to 2-oxyglutarate in the TCA cycle is extremely near. Lysine, as we'll see, that hasn't changed, but others, kyuranine, oxoadipate, 2-aminoadipate, have gone down very significantly in abundance in these mutant cells. And so one of the things we did was we want to understand what's happening to the metabolic system as a result of these mutations. And just for these examples, an interesting thing pops out of this, and we'll come to some other analysis of the data, but if we just look at lysine degradation, so a number of the metabolites that were significantly depleted, here's an example, saccharapine, are found in lysine degradation pathways. You can see there, saccharapine is produced from lysine via incorporation of 2-oxyglutarate, 2-OG. 2-oxyglutarate, if you remember, is the metabolite that is converted to 2-hydroxyglutarate in mutant cells. Also, we've got 2-aminoadipate. 2-aminoadipate is depleted significantly. And you can see down there, it's a little bit further down in lysine degradation. Similarly, 2-oxoadipate is the next metabolite that's 2-aminoadipate is converted into, and involves the 2-oxoglutarate. And this is a cofactor, so 2-oxoglutarate is being converted to the amino acid glutamate in that process. So it's not, the carbon atoms are not being incorporated into 2-oxoadipate, but it's required for the reaction to take place. We've also got here the tryptophan degradation pathway. And here's kynurenine, which is formed from tryptophan. And kyurenine is converted to kyuronic acid, again, via 2-oxoglutarate. And indeed, 3-hydroxykyurenine is converted to xanthurenic acid by 2-oxoglutarate. So there was a theme that came out of this discovery-led analysis, this metabolomics work, that we hadn't anticipated. But it became clear that what's happening is the depleted metabolites, the significantly depleted metabolites, are predominantly occurring after reactions that involve 2-oxoglutarate as a cofactor, or indeed as part of the reaction in some cases. And so we think that this is important because 2-hydroxyglutarate that's produced from the mutant reaction, the change of function mutation, is extremely similar structurally to 2-oxoglutarate. And one hypothesis here is that what's happening is that 2-hydroxyglutarate is causing substrate inhibition of these reactions, and therefore stopping the conversion of some of these degradation pathway reactions. So how do we go from a sort of bigger, what we want to do is go at a bigger scale from these features, these compound features, to function. We got a little taste of that with those degradation pathways. And so what we do is to take these features and use pathways analysis, not just to pick out particular changes, but to map all of the metabolite changes that we can identify in the metabolomics analysis onto metabolic pathways to find which of those have been altered and which ones have been altered, to what extent they have been altered, which is what is represented here on the right-hand side with this metabolic pathways analysis map, which really shows the extent, the significant change in a metabolic pathway now on the y-axis, and the enrichment, the amount of that impact, amount of altered metabolites in that pathway on the x-axis. So things in the top right are going to be where you've both got high significance in the data, but also a significant alteration in those pathways. And you can see the TCA cycle, pyruvate metabolism, C5 branch dibasic acid metabolism up there, and several others that I haven't labeled at this point, showing a sort of that there is, well, the key thing to take home from this is that there are a number of metabolic pathways that have been altered in IDH mutant cancers as a result of this change of function. And another way to look at that data is to through the lens of these reactions that involve 2-oxoglutarate that we saw for the lysine and tryptophan degradation pathways. So sorry, this is a bit of a blurred image, but it just crams together most of the metabolic pathways you find in mammalian cells. And what I've done is to highlight in yellow those that involve 2-oxoglutarate in reactions, and overlaid to those with in red where those pathways have been altered significantly in our pathways analysis. In green, we have pathways that don't involve 2-oxoglutarate but are also altered significantly. And clearly you can see a lot of amino acid metabolism has been altered, and a lot of amino acid metabolism has 2-OG dependent reactions in it. And so we think that this comes back to as illustrated by lysine and tryptophan degradation, is likely to be the reason why these are altered, is likely to do with 2-HG inhibition of substrates. And intriguingly, other non-2-OG dependent pathways that are important in energy transduction, antioxidation, there's glutathione there as well, and also nucleic acid metabolism, purine and pyrimidine metabolism, significantly altered too. So yeah, this is for us was a really nice place to get to from trying to asking those questions about central metabolism and coming up with a whole range of altered pathways. Clearly important, we believe, in tumorigenesis. Okay, so and work is ongoing there. One of the things I've not shown is work that we've done to complement this with transcriptomic analysis, because some of these pathways, we believe, aren't necessarily, some of these pathways look like they are directly being affected by 2-hydroxyglutarate levels, as in enzyme inhibition, but also other pathways appear to be altered where 2-HG may not, 2-HG or 2-OG is not directly involved. And so transcriptomic analysis has actually shown, and I won't show this data, but just to point out that we've seen significant changes in transcripts associated with metabolism as well, and matching those to the metabolomics analysis is work that's ongoing. So I'll leave the IDH mutations there as an application. And in the last part, I just wanted to go on to talk about some work that's ongoing at the moment. This is unpublished work now, work that we've been doing to look for biomarkers in the context of early detection of cancer associated with hepatocellular cancer in this case, using anion exchange chromatography coupled to mass spectrometry again. And in this case, rather than looking at cells, we're looking at patient urine samples. Isabel and Mia, both in the group, have been working on this year. And we're in collaboration with Jeremy Tomlinson, who's works on hepatocellular cancer work here in Oxford as well. And so this has really driven by the question, can we see metabolic biomarkers that differ between those with cancer and those without cancer? And what do those look like? So what we did was to take 40 healthy controls, urine from 40 people who were healthy, as far as we understood, those that have got HCC, hepatocellular cancer, and those that didn't have cancer but had cirrhosis of the liver, which increases the risk of developing HCC. And it should point it out that those with hepatocellular cancer also are likely to have liver inflammation and cirrhosis as well, for that reason. So we took 40 of each and analyzed those on our ICMS platform and had a look at the results. Oh, well, actually, just before we did that, we did some testing. So we wanted to find out what are the best sort of approaches for preparing those samples, and for urine samples. We hadn't worked on that before. And so we did quite an exhaustive, or Izzy and Mia did quite an exhaustive analysis of different extractions and filtrations of those urine samples using molecular water and acetonitrile, methanol, butanol, different types of solvent, also with and without molecular weight cutoff filtration, and essentially, there's no magic. There's no particular one of these that is a lot better than others. I think the key thing to come out is one of the benefits of ICMS, which is you could use pretty much any of these. There are some, but I'm not going to go into the detail of which is a little bit better than others at this stage. But molecular water, 80% molecular water works quite well, actually, as well as methanol extractions too. But this work was useful to sort of validate. And going forward. So when we did the analysis to compare hepatocellular cancer and the healthy controls, the first thing that was really clear was that there was a significant difference, both in the PCA plot on the left-hand side there, as well as the sort of top compound features that were altering in abundance. There was a clear range of compounds that were altered in those with hepatocellular cancer versus healthy controls. And so this was very interesting for us, implied that there were metabolites, that biomarkers of these differences between these groups. And unpublished, as I say, so I'm not going to actually reveal the metabolites today. We're working on this at the moment. But this is a volcano plot showing just the identified compounds that we measure. So we measured 293 metabolites and identified those. 37 of those were statistically significantly altered in hepatocellular cancer patients, FDR corrected at 0.005 and 1.5% fold changed. So a number of metabolites, 21 of those were downregulated, 16 were upregulated in the HCC. And so this is very interesting for us. We want to know we were identifying what those are and trying to see why they were altered, I think is one of the questions we're looking at the moment. Interestingly, when we overlay the patients who have cirrhosis but don't have hepatocellular cancer, and those are the ones in green, you can see that overlaps very significantly with those with hepatocellular cancer as well, but are distinct from the healthy controls. And that's perhaps not surprising, as I've said, those who have hepatocellular cancer also have cirrhosis and inflammation, or most likely to. And so that's what we saw. Intriguingly, we did see some metabolites that differentiate those with cirrhosis and HCC, but the majority of those overlapped. So what we think this shows, actually, or this analysis is showing, is something focused around liver function and liver condition than necessarily hepatocellular cancer biomarkers, although, as I say, there are a few that may be related to that. And I think this is interesting and useful because what it shows is that we have a method here that is giving us a kind of direct readout, we think, of liver function, and that's important from a disease perspective. So early days, we haven't completed this work yet, but it looks like anion exchange chromatography is able to or show that there's a strong phenotype in hepatocellular cancer patients from their urine samples and those with cirrhosis as well. 37 of those metabolites are significantly altered in HCC. And indeed, this includes biomarkers of HCC that we think, a few, as well as liver inflammation overlaid. And I haven't shown that here, but we are able to use multivariate modeling to distinguish these HCC and cirrhosis groups from healthy controls as well, which is useful for predictive modeling, would be useful hopefully for patient screening. Okay, I'm going to leave it there. That's the leave you with a few publications from the group over the last 10 years or so, where we've used ion exchange chromatography in an omics context, particular metabolomics. And we're finding it, well, hopefully the examples have shown that we've found it particularly helpful as a tool for focusing on primary metabolism, the primary function of producing energy, of producing biosynthetic building blocks for cells. So just to conclude the last couple of slides then, so we've shown metabolomics is analytically challenging. We knew it was analytically challenging, and it still is. There's no single method that provides comprehensive coverage. ICMS doesn't provide comprehensive coverage either. It focuses on highly polar and particularly ionic metabolites. But we think this is really useful when it comes to changes in primary metabolism. So one of the things I think that ICMS clearly offers is the robust and reproducible analysis and the ability to capture highly polar, highly ionic metabolites. And this is important because these are really important metabolites in many, many contexts. It outperforms Hilic MS in that context. And I think the other thing is really the robustness is partly because it's very compatible with a wide range of sample preparation approaches. You can have fully aqueous, you can have fully organic, and the system works well with that. And we have shown that we can transfer that information, those signals that we get back from ICMS, into a systems biology workflow that gives us information about functional biology. We can use it in targeted as well as an untargeted context, and in fact, the untargeted context has been where we've developed some of the most important things, I think, from a discovery perspective. I haven't focused on them too much, but there are challenges. I mean, the low polarity compounds we don't see by ICMS as we might expect, and so we do need complementary techniques. I didn't mention in detail, but Zwitter ions. So if you remember right back to the beginning where I talked about the ion suppressor taking out counter ions. So if you've got Zwitter ions, and key example are amino acids in metabolism, those will be taken out by that counter ion suppression, and so we don't characterize those well, other than a small number that the acidic amino acids, glutamate and aspartate. And the other thing that is somewhat challenging is that the chromatographic resolution is relatively broad compared to, for example, UHPLC, because we haven't got really high pressure systems here, and so peaks are a bit broader than we're used to for UHPLC. But these are challenges. These are things that we were working on. We're working on in our group in all of those areas, and I think that they're problems to be solved and will be solved. So it works really well, as I say, for primary metabolism, and I think this is the key area that we work on. We've shown, I've given examples of human biology today, but we've also looked at microbiota and plants as well. And just to finish off on this theme of central metabolism, so the most abundant metabolites in mammalian cells are negatively charged and are involved in primary metabolic pathways. These pathways of energy transduction and biosynthetic building blocks to make new cells. And I think this is why ICMS works really well, because those are so key to cellular function that when things go wrong in cells, it's these pathways that are actually altered. They're conserved pathways across different organisms and are highly important in plants as well as microbiota and humans. And the pathways, glycolysis, TCA cycle, oxidative phosphorylation, amino acid metabolism, lipids, nucleic acids, redox metabolism. That's a key area, I think, that we focus on less, but is very important in disease. These are all disease-modified pathways commonly. And it doesn't matter whether it's infectious disease or non-infectious diseases, and indeed rarer inborn errors of metabolism. These all tend to have quite significant driving changes in these central pathways. And I think, therefore, ICMS has a really important role to play in looking at those, using in an omics science application context to look at these pathways. And has a real contribution to make to metabolomics and omics science. Okay, I'll just leave it. I think we'll finish there, and to say a big thank you to everybody in our group who has contributed to ICMS over the last 12 years, as well as our collaborators, many of whom, some of whom I've put here, but many of whom have been involved in other projects I haven't talked about today. Also, a big thank you to Thermo Fisher Scientific, Susie Bird in particular and her team, and Wai Chi as well in the UK, for supporting our research. And to thank you all as well for joining us on this webinar today. Thanks very much.
Thank you, James, for such insightful talk. And you know, great to hear about what you and your team do. And we look forward to seeing more of your fascinating publications from your group. And we have a few questions that's coming.
Sure.
So hopefully we can get those answered. And what's the first one? I think everybody agreed that it's been a very insightful presentation. Can you comment on how IC performs relative to ion pair chromatography?
Yeah, that's good.
Ion pair methods offer stable retention. The resulting system contamination limits the instrument to ion pair applications only. And the helix is not stable. We are looking for alternative methods.
Yeah, that's a really good question. I can't see the question myself, but from what you've said, the comparison between ICMS and ion pairing, we've done some work with ion pairing early on. One of the challenges. I think that the person was commenting, sorry, I didn't get the name, is that iron pairing reagents often contaminate the system that you're analyzing by LCMS. So many iron pairing reagents lead to iron suppression in other iron modes. So if you're looking at negative, then your positive iron mode will be stressed. I mean, iron pairing works quite well, I think, but you need to have a dedicated system for it. And the nice thing about ICMS is that you don't have any of that. There are no iron pairing compounds. And again, you can really look at extremely polar metabolites, these triphosphorylated compounds, which we found was particularly helpful here. So we don't use iron pairing because of the contamination issue, but I think it's an interesting and useful option compared to Hillit. I don't know if that fully answered the question, but
I'm sure if we need to sort of explain more, we can always reach out afterwards. But please just reminding everybody, you can submit your questions via the Ask Question widget pinned to your console. And hopefully we can answer those questions on this call. If we can't answer it, we will answer it offline via email to you. Okay, and what are cancer-specific biomarkers for early detection and how metabolomics helps in this?
What are early biomarkers for cancer detection, and how does it help? Good question. So I think well, the IDH mutation leading to changes in 2-hydroxyglutarate. This is a really good example of an oncometabolite, 2-hydroxyglutarate. An oncometabolite being a metabolite that changes significantly in abundance and is kind of characteristic. It's led to the World Health Organization. It's led to the way in which these cancers are defined, being changed to incorporate these mutations. It's unusual, though, to have very big changes like you see for 2-hydroxyglutarate, and therefore, I think biomarkers of cancer are not necessarily, we shouldn't necessarily be looking for individual compounds as being a biomarker, but rather, I think what's clearer, and what came out, for example, of this urine analysis, is that there's a range of metabolites that have altered, and they provide a sort of together provide a profile that is telling you about the biology of the tumor. Now, the challenge is, of course, when you have a very early tumor, you have very little of it. And so we're not quite so clear as to whether we're picking up changes that are directly as a result of tumor genesis, driver mutations leading to changes in metabolism, or whether or not there are changes in the microenvironment that take place before those tumors really develop. And it comes back to sort of fundamental questions that are encapsulated, I think, in the IDH story. You know, there are driver mutations that are, and there are mutations that are clearly very early in tumor development, but to what extent do you need that microenvironment around those for those to really drive tumor development? And it may be that 2-hydroxyglutarate and some of these other metabolites are helping to drive that microenvironmental context that allows tumors to grow. Because there are many processes that are, obviously try to resist these tumor development. That in terms of reducing cell proliferation, etc. These have to be kind of removed by mutations as well. So yeah, hopefully that gives a bit of an answer to that question.
Okay, and what are the key ways metabolomics data can be integrated into the drug discovery pipeline?
Yeah, great question. I've been thinking about this quite a lot, actually, as well. So, yeah, I'm not sure. There's a long answer to this probably, but I think the short answer is I think we're still trying to work that out. The drug discovery pipeline has evolved. It is very expensive to make a new drug, as we all know. And essentially, it started as a trial and error process, right? You take a lot of compounds and you screen them against your targets to find those that kind of that work, that hit the target. The problem is that you have to screen millions of compounds, and that once you develop a drug, it takes 10 to 15 years and billions of pounds, right, for a cancer drug. They don't always last very long. You get resistance to drugs very quickly. So the context is challenging. Sorry, I'm paraphrasing that obviously quite a lot. But so ways in which we can use metabolomics and other omics sciences to try to drive that process further, I think are really developing now. And those really revolve around trying to understand a broader context, not just one hit on one target, but how do we prevent, in the context of cancer, for example, how do we prevent tumors developing by understanding how their metabolism is altered to support tumorigenesis? And this is really where we're focused. How do you target not just a particular mutation, but how do you target a range of mutations, but also a range of tumorigenic, of metabolic processes that are resulting from that to support the tumor development. And I think IDH is an interesting example where we see one mutation leading to lots of changes in metabolism in multiple pathways. And the key question is, which of those are vital to tumorigenesis, which of those are incidental? And that's what we're working on. And I think once we have ways of using omics science to identify, and I don't think it's just metabolomics, it's incorporating transcriptomics, proteomics, and metabolomics into analysis. Once we understand that much better, I think targeting multiple sites, multiple processes that prevent cell proliferation from immortalized cells is the way forward. Not easy to do, but I think we are moving towards that.
Excellent. There's a question that I get asked quite a lot. Doesn't the hydroxide damage your mass spec?
Yes. Well, it would. But this is the key thing here is that the ICMS system has this ion suppressor that's built in after the chromatography, but before the mass spec. And it's designed to electrochemically neutralize the hydroxide that's present in the mobile phase, which you're right, if that went into the electrospray source, the ion of the mass spectrometer, it wouldn't last long and you'd have major problems. But the neat trick is that you are neutralizing with protons that hydroxide so that you only have water going to the mass spec, and water is fine. And actually, the modern electrospray systems can handle highly aqueous mobile phases going into the system very well. So you've got a choice. You can tee in an organic solvent if you like, or you can just leave it as an aqueous stream. And we find that the aqueous stream in many contexts works very well with electrospray. So the answer is luckily not.
And just time for just basically one last question. What is the greatest impact ICMS has made in your lab?
Great question. The greatest impact it's had. I mean, I think I tried to encapsulate that today in the sense that it was really it's really transformed our ability to characterize highly polar and ionic metabolites, highly ionic metabolites, which were really difficult before. And I think the transformation, the ability that it has to do that has really transformed the way in which we can access primary metabolic pathways that we were capturing to some extent previously, but it's really been a step change in the ability and the fidelity that we have to do that. And that has led to us finding things out in a whole range of contexts, cancer and non-cancer, particularly around disease metabolism, that has been really great for us and has led to understanding how primary metabolism is altered in these disease states and our biomarkers in ways that we just wouldn't have had access to with other methods.
Excellent. And we also have more thank you messages from our participants. Thanking you for a valuable and thought-provoking presentation. So that is always good to hear. So basically, that is all the questions we have time for, because I want to sort of basically ask you the very, very last question, because we're celebrating 50 years of ion chromatography innovations. Where do you see IC technology going in your work, and how can it be more impactful?
Where do I see ICMS going in the future in our work, and how can it be more impactful? Great question. Good question. So, I mean, I think, yeah. So, I mean, we're still developing the methods, right? So we've got a really nice platform we've published, but there are challenges that I mentioned. For example, these amino acids, the resolution. We would like those to be, those are problems we'd like to solve with ICMS. We think fundamentally ICMS is a great platform for omics science, but there is probably more we can do with it. I don't see us. I see us having ICMS as a key platform in our omics going forward, but we'd like to solve some of these challenges, some of these kind of aspects that will just improve its capabilities. And I think that's what we will be doing over the next years, I hope, and working on those particular problems. So that's what we'll do.
Thank you very much. So, thank you to all of you have joined us celebrating 50 years of ion chromatography innovation. And just like to sort of mention a few bits that we will be end today's webinar and provide you, hopefully you can provide us with your feedback in the short survey widget on your console. The resources are available to download in the related content widget. Lastly, a recording of this webinar will be sent directly to you via email within 24 hours of your for your convenience. So thank you for your time and your attention today. Thank you very much.
Thank you.
Have a lovely day.